This is for assignments 3.5A and 3.5B. Those assignment grades will go on the next 6 weeks. The tissue grades, where you studied each of these tissues, that's multiple grade. At least 1 grade for each of these types of tissue based on your table. Reading different genes means making different proteins. We have an organ like the human liver, and we know that, okay, there's some kind of genetic code that we get from our parents that ensures we get a human liver and not a cow liver. Not only that, but the specific code from our mom and our dad, the whole code is in one cell to begin with. That's you, just one cell. But how do you get from that one cell and the entire code to all the specifics that make up, say, your body organs that all work together for the function of keeping you alive and hopefully happy? So, cell specialization. Specialization. What is our topic? Well, anatomy and physiology, structure and function. Okay, but how do we come up with all the structures that make up the human body and all its many, many, many functions that are very specialized? So cell specialization, that's the what, is this particular thing. And this is your first diagram. It was like this. And it represents how we go from a blank cell or an unspecialized stem cell, or in the case of your baby, being a baby, starts out as a zygote. And these stem cells that divides and becomes stem cells, it could become anything. They're pluripotent. They're potent. They're potentially anything, any specialization. And how do they go through this process? So to become the 4 types of tissue. You know it's a how process because you got all those arrows. Then we got a how diagram that was an on-off process. Genes get turned on and off. So we just have the genes based on cues or factors. In this area, it's like unbalanced in different parts of the cell so that it's the focus that gets put in front of you. It's the cue. It triggers your attention. Just like I've got a bright color, so it would get my attention and I would get my work done. So there was a cue, these factors, that led that cell that divided to become more muscle tissue-like. So it did that by turning these factors cued for certain genes on the DNA to be turned on and to be making proteins, and other genes to be turned off. So we're not making those proteins. So as we go down this way, we're seeing that genes are getting turned on that produce muscle proteins that would function for muscle movement, and nerve type tissues or nerve type proteins being turned off so that those aren't formed as it specializes, until finally we get very specialized and very specific. This is a cardiac muscle cell, not a skeletal muscle cell. So you can see even within these, they're similar compared to these other types, but it's more specific, and that's because of the environment. The environment. There are cues along the way so that its environment eventually, when the muscle tissue is in the heart area of that baby body, then it's gonna just turn on the genes. That environment will turn on the genes that make only cardiac muscle tissue, rather than like the skeletal muscle tissue that moves your skeleton, that pulls on bones. And so there you go. In the area of bones, muscle tissue will specialize as this type, and it will have the specific proteins for this type, and so on, smooth. So the same muscle cells that are in like the stomach area, or your swallowing muscles or something, or not your swallowing, but your esophageal muscles after you swallow, that squeezes your food down, that you don't really control, then in that area they're going to be smooth type proteins. Okay, now we've got nervous tissue direction over here, just as we had muscle tissue here, and of course they can specialize into neurons or glial cells. And connective tissue, we have an example of red blood cell. I didn't want to try and draw all the other ones for connective tissue, even though that's most, and its functions in general are providing support. What's this one? Can you guess? Can you remember? What type of tissue? It is epithelial tissue. And so we have an example, waterproof skin. So the proteins that would do things that make it waterproof are embedded in that skin. They would be cuboidal probably, and producing oils that waterproof it. Also we have stratified squamous, these flat stacking pancake layers, so lots of layers, so protects you from the many abrasions that you might get or cuts you might get. And so we can see the environment in that first cell is how it will eventually could become any of these types of tissues eventually. And this is much like, as we said in class, much like your environment shapes you and affects what you will become in your future, and how productive you will be, how specialized you will be, what kind of a job you can do, if you will put in front of you yourself and in your environment something that will benefit you in that way and give you that direction. If I just float around, then who knows which direction I go. It may not be a direction I want to go. But if I am intentional and I put in front of me, I'm gonna use my favorite pen to do this work, that's gonna give you a little environmental direction toward focusing on what you need. Your assignments are, of course, to do those diagrams I just showed you, but also to watch that video again, put that video in front of you so that you can explain in 10 sentences how do cells get specialized? How do they go from one to any type of specific cell in a specific organ and make it whale money?